I'm working here in Hanover. I'm the director of the Institute of Zoology at the University of Veterinary Medicine here in Hanover. And we are working on the behavior and on evolution of animals, especially on primates. And one of the primates you can see here on the computer screen, it's the mouse lemur here. It's the smallest primate in the world. It has about the size of an apple or an orange, and it's the smallest primate species which exists nowadays. So in Alzheimer's disease patients, one of the most obvious signs of Alzheimer's disease is a loss in learning and memory, which disrupts the daily life of Alzheimer's patients quite strongly. And Alzheimer's patients, for instance, they forget what they have learned recently. You can measure these deficits in learning and memory by a computerized cognitive testing tool called CANTAP. When a subject fails to perform, for instance, specific tasks, you can characterize then the affected brain region, and this helps in diagnosis.
So this is done for humans?
This is done for humans. And you can transfer the methods, or we are about to translate the methods which were established for humans and for the diagnosis of Alzheimer's disease to the non-human primate model mouse, to the primate you see here. So we established a computerized cognitive testing tool called the BASI Saskita touchscreen chamber, and cognitive tests developed for humans and adapted it to the non-human primate model. And the goal is to disentangle healthy brain aging from pathological brain aging in them, so that we have comparable experiments in humans and in non-human primates, and can then conclude that brain deficits in primates correspond to brain deficits in humans. Mouse lemurs can be trained to indicate us by touching on the computer screen if they have recognized a specific task or not. So you can get some insight into learning and memory by testing them by a touchscreen experiment on the computer. For instance, pair discrimination tasks. So you show them two different images on the computer, and only one of the image is correct, and they have to indicate that by touching towards the computer screen. Then they get a reward. So you can test by doing that two-choice pairwise discrimination, learning and reversal. You let them learn, for instance, at first that a circle is the correct image they have to touch, and when they have learned that, and you can establish that by a specific learning curve, you display, for instance, a quadrat as the correct signal, the correct stimulus. And then they have to reverse the learning. And this reversal learning is a bit complicated, as you can imagine. For instance, from humans, we know that Alzheimer's patients or Parkinson patients have problems in doing so. And we have tested that in mouse lemurs, in young mouse lemurs and in older mouse lemurs. And mouse lemurs did their task quite successfully. But we get interesting learning and memory differences between youngsters and oldies. Youngsters learned their task quite rapidly, and they do also the reversal learning quite rapidly. Some of the oldies had great problems, so they learn much slower. And some of the oldies had also problems in the reversal learning. So it means that they are not as flexible as youngsters are. when you use a primate model for doing research, there are specific regulations. So we are working here in Europe. There is a European law for the protection of animals in research you have to consider, and especially in Germany the situation is quite strict. There are quite strict regulations governing research on laboratory animals. So there are guidelines worked out for the care and use of animals in research. There is, for instance, also the Amsterdam Protocol for the protection and use of animals in research, and it's quite necessary to have these different laws and regulations, so to guarantee that these animals are treated quite well, also by researchers. You have to apply for permissions to doing research, even behavioral research, also in the European Union or in Germany. We have done that. We have a quite nice facility for breeding the animals, and we have standardized experimental protocols for doing the behavioral research. And we applied for these licenses and got them. So these licenses rule that everything is quite well treated with regard to the protection of the animals and with the care and use of the animals in research. We hope that we will identify early precursors of Alzheimer's disease, since we have a so-called cognitive model here, in which we can quantify learning and memory deficits on the basis of using these non-human primate model. We will establish non-invasive methods to quantify also brain morphology. So you can use magnetic resonance imaging for gaining a picture of the brain in the living animal, so that you can detect also brain pathologies or like atrophies, for instance, in the brain. And you can then link results on the behavioral level, on the cognitive level, with results on the brain morphological level and with the underlying genetics, to be able to identify then early precursors of aging diseases like Alzheimer or others. Aging diseases are of high concern, of economic concern also in the whole world. And it's quite necessary to detect diseases, brain diseases, at the earliest possibility, at the earliest age possible, to then also treat the respective patients, so that you can avoid very high economic costs which are underlying these diseases when they spread. Since the human population will get older and older as we see it nowadays, and insofar you have to prevent aging diseases as early as possible.